sequences. Okay. Thank you for the introduction, and also thank you for the community to invite me to give a talk. So I want to tell you a different story, starting from RNA-seq. So probably you know that RNA-seq is nowadays the most popular method to study the transcript. Many people do RNA-seq because it's very cheap, and you can just send the sample to the company and you'll get data. For the RNA-seq, so briefly, it's just break the transcripts into small pieces and then do the sequencing, right? So then after you get the data, you can map all the reads back to the human genome. Then you know the expression of all the human genes. Since we are in the L community, then you definitely know that in the human transcriptome, we also have transcripts coming from T-cell receptors and B-cell receptors. The problem of those transcripts is because there is a rearrangement in the CD3 region. So that's why normally when people try to map the reads that are coming from the CD3 region back to the human genome, it's unmappable, right? Then you can guess what people do to those unmappable reads. They throw it away, right? So then you can think it's like a loss. It's just a waste of information. So that's why in our group, back to 2016, we try to recycle those T-cell receptor and B-cell receptors from bulk RNA-seq data. So the first paper was published in Nature Genetics, which focuses on the T-cell receptors, and we scanned through more than 9,000 tumor RNA-seq samples in the TCGA cohort, and then we can identify millions of T-cell receptors. Earlier this year, we do the same thing, just scan all the B-cell receptors. Very surprisingly, I think because the plasma cell have very high expression of their transcripts, so we can identify 10 times more B-cell receptors. So it's 30 million B-cell receptors across 99,000 tumor samples. Meanwhile, the MagCell group also extended their algorithm to handle the RNA-seq data. And we noticed their publication, and you know that when people want to publish a paper, they compare with existing ones. And after reading their paper, we realized that there is a big disagreement in how we measure the performance of this whole recycling process. So I'll explain to you. So actually, because there are so many details, technical details, we actually publish all those discussions in the Nature Biotech. So I don't want to bother you about all the technical details, but I want to emphasize there is a key disagreement between our group and the Maxima group is how we define the sequence that is recyclable. Okay, so because you can imagine you get all this random sequence from the RNA-seq data, what is useful, what is not is a big question. So to make it more specific, let me give you an example. So we did AIRR-seq for tumor sample. Then, for example, we just do clustering, and then we can find a small cluster with 7 CD3 sequence. They are very similar by looking at the alignment. So you can clearly see that they're coming from the same source. And you can see the somatic hypermutations, right? So if we take the same tumor sample and then do RNA-seq and then run those recycling algorithm, what we can get are those sequences I highlighted in black. So you can see for the first one and the third one and the 7th one, we can actually recycle the complete CD3 region out of the RNA-seq data from the same tumor. The tricky part is you can see for the sequence 6, for this sequence, we cannot recover the whole region because of the randomness in the RNA-seq data, right? It's randomly chalked. So, however, we can recover a majority of the CD3 region. So that's why we try to define this kind of situation is a partial CD3 sequence. And we try to do research based on both the complete CD3 and the partial CD3 sequence coming from RNA-seq data. So This some of the figures in our paper. So if you're interested, you can read the paper. So the first thing we check is the somatic mutation. So because like for those tumor infiltrating B cells, they were already fighting against the tumor for a long time. So you can imagine their receptor is quite mature, right? So that's why when we compare the somatic mutation pattern, it's different from other cases. Is you can see there are lots of like synonymous mutations other than non-synonymous mutation, meaning that they don't have much room to change their CDR3 sequence because it's already like staying there for a long time. Also, because we have the somatic mutation pattern, we can somehow infer the somatic mutation rate, and across the thousands of samples, we can also load the AID expression from the RNA-seq data itself, right? Then we can see a very strong positive correlation between the somatic mutation rate and the AID expression. Similarly, just the quality control, we also group the CDR3 sequence based on their isotype, and we can find there are more mutations in IgG consistent with what people have found in other cases. Since we are dealing with TCGA data, which is a wonderful data set, have very detailed clinical annotation, so it can give us the power to compare what's the difference between the tumor and the normal in terms of the B cell activity. And clearly we can see that there are more B cells in the tumor samples, and also there are more like IgG, specifically like the switch from IgG3 to IgG1 across multiple tumor types. Of course, in terms of the somatic mutation rate, they are different across different tumor types. We can see, like for example, for the lung cancer, they tend to have higher somatic mutation rate. Probably it's like a kind of hot tumor. Also, we try to like do some analysis by combining the somatic mutation rate and the IG switch information. So we have 4 cases, right? And clearly you can see that for those patients who have both high somatic mutation and high ratio of IgG3 to IgG1 switch, they tend to have better survival compared to other cases, indicating that overall, if you have more B cell activity, you tend to have better survival. Of course, we cannot tell you like the mechanism, but this is what we observe across thousands of samples. The last part of the story is we also, for the main function, one of the main functions for antibody is the ADCC, which is like the antibody when it recognizes the tumor and it will recruit natural killer cell, and the natural killer cell will kill the tumor. Okay. However, their tumor cell find a way to evade the ADCC attack through upregulating the MYC-A and the MYC-B gene. So it's a long story. Like when they upregulate it, there is some enzyme to chop the MYC-A protein into a soluble MYC-A MYC, and then this soluble MYC will bind to the NKG2D and internalize the NKG2D and deactivate the natural killer cell. So this whole pathway is well understood like for 10 years. And what we found is we checked the MYC-A application and we checked the isotype, and we found that for the patients who have MYC-A application and the high antibody activity, they tend to have better survival, meaning that there's some protective antibodies. Yeah. So I think after I show you all those things, I hope I can convince you that those partial CDR3 sequences are useful because it can help us to check the somatic mutation and ID switch pattern. And also, I think this is useful because it's useful for machine learning, right? I copied this slide from Professor Cohen's presentation yesterday. So in her model, those CDR3 sequences are chopped into sleepers, right? So if you use sleeper, then it doesn't matter whether it's a complete one or partial one, right? Of course, the caveat here is that you need to have a special algorithm, special pipeline, special like methodology to evaluate the performance to do computational things. So I think I forgot to tell you like the position. So last month I was a postdoc at the Farber Cancer Institute, and now I joined a startup called the GP20 Therapeutics, which is focused on the antibody drug discovery. So in our company, we have those like experimental platform and the computational platform, which try to use those recycle all those informations from public domains and try to find those therapeutic antibodies from data. So I would like to thank my mentor at Dana-Farber, Professor Xiuli Liu, and Professor Bo Li, who was a former postdoc in lab, who designed the trust algorithm, and now he is an assistant professor at the UT Southwestern. Okay, thank you.
Thank you. Okay, we already have questions.
Okay, so that is very impressive analysis of immunoglobulins inside a tumor. But there's this fundamental question: how do you know that these sequences have some relevance to the cancer? Like maybe these are recruited B cells, plasma cells, which make antibodies against something else, or some polyclonal. How do you know that they are really relevant for the cancer?
Yeah, so like for the MICA case, we somehow guess it may be an autoantigen because there were already like a drug which derived from like patient anti-MICA autoantigen, autoantibody make a drug, right? So there are examples like this. Also, like for the many tumor, like for HER2, and there are many known like tumor antigens that exist. So we assume that sometimes they can recognize those antigens, tumor antigens. Sometimes maybe it's autoantigen, right? We don't know. Then yes, that's what we are doing using the deep learning. So we have using machine learning method. Yes, yeah. Of course, as I show, that's one of the reasons why I joined the company, because the company we also have an experimental platform to validate what we predicted from the models. Yeah.
Okay. Thank you. Any more questions? Okay. Oh.
I have one question. That was a very nice talk. I just wanted to know if you found any correlation between the tumor mutation burden and the somatic hypermutation.
Yeah, we checked it in the first Nature Genetics paper. That's it for the TCR. For the BCR, we didn't check that.
Okay.
So if I recall from this controversy that erupted a year or two ago, it came down to accounting of those partial fragments, whether they were distinct clones versus
Yes.
they should be grouped with
Yeah.
the full-length sequence to represent one clonotype.
Yeah.
So I'll just ask the same question that I was asked earlier, and that is, would you consider CDR3 to be a different clonotype and have different potential antigen recognition properties if it's combined with different V and J segments?
Yeah, I think this kind of like discussion or question is relating to how we measure the model performance, right? How we measure, you just define some metric and then you can get a result, right? So different people have different opinion on how to measure the accuracy, specificity, and so on. So we didn't hide any we didn't use it. We have our specific definition of accuracy, specificity. We agree, but we didn't hide it. Because it's a special data. For most AIRRC data is complete. So I think people agree there are some standard way to compute accuracy and specificity. However, for those partial ones, we don't know. We don't have a reference to do the right computing, right? So that's why I don't think it's a big problem as long as we tell people that this is the way we're doing the performance evaluation. And also, I think finally, we want to reduce all those complexities in a machine learning model. Because for the machine learning, you can imagine when people do image recognition, they try to change the image, right? Chunk it into pieces, and then rotate it, which give a better result, right? The same thing here. I think we need to somehow those partial ones are informative because it's like an image. If the model can give a good prediction, even for those partial information, meaning that they are recognizing the right paratope information in the antibody sequence. Yeah.
Okay. Okay, at this point, we've got to move on. So thank you, Dr. Hu again. And